Hello students, welcome back to your science class. So student, in the previous class we were talking about the asexual reproduction that take place in the plants, and yes, we know that the plants, they used to show the sexual reproduction. The angiospermic plants, so the flowering plants are there, they consisting of the male and the female gamete both, and that is present in the pollen grain, and that is present in the ovary, and this will fuse together, and due to which there is a formation of a zygote occur, and due to which the formation of a seed happens. But it is not only that every time the flowering plant is there, means not only the sexual reproduction take place, but the asexual reproduction is also responsible for the parts of the, for giving rise to a new plant, and this asexual reproduction occur from any of the part of the plant. Like we can take a stem, or we can take the bud of the notches which is present in the potato. And in the last class we have studied also that how the buds which is present in the potato, or how the flowers which is present in the bryophyllum leaves, they get detached. You know that at the edges of the bryophyllum the flowers are present, and these flowers get detached, and when it reaches to the soil it will grow into a new plant. So basically we can say that the leaves of the bryophyllums, they are responsible for giving or for the development of a whole plant. So it help in generating a new plant from the one of the part of the plant. Okay, similarly in case of a potato also we have seen the bud. Even we have understood that if what will happen if suppose when we plant a piece of a potato which consisting of a bud, and a piece of a potato which do not consisting of a bud in it. So what happen when we do that, the piece of the potato which is having the bud in it, it will it will get germinate, and the growth of the root and the shoot will take place from it. But the piece of the potato which do not consisting of a eye in it, the consisting of notch in it, the consisting of bud in it, so obviously that will not going to grow into the root and shoot of the plant. So yes, there is the condition also that what are the important thing, even if we are taking any part, how we are using that part in order to develop a new plant from it. There is another method, student, which is the latest method you can say, and it is important that tissue culture is what? So tissue culture is what, student? Tissue culture is basically a new technique by which we can generate a new plant from it. And yes, why we are saying that it is a new technique? It is because this first of all, see here, it is written here that in tissue culture new plant are grown by removing the tissue or separating cell from the growing tip of the plant. Actually what happened here, we will take the small tissue by separating the or we can say by or we will take the separate cells, or we will use a tissue, and from the growing tip of the plant, because we know that the plants are what, you are very well aware of the different types of tissues, students, and we know that one of the tissue which is the plant tissue called meristematic tissue, and they are what they used to grow, they basically present at the tip of the plant, tip of the root and tip of the shoot also, and they are responsible for the growth of the plant. So if we will separate few cells or the tissue from that of the growing tip and use that, it means when we will take this and keep it in artificial medium, the medium where we are going to provide the sufficient the nutrient to them where they can easily divide, and when they will rapidly divide, they will form a small group of a cell, and this small group of a cell is also called as callus. See here it is written, it is also called as a cells or callus. Now this callus is transferred to another medium, that medium which consisting of hormones for growth and the differentiation. Yes, afterwards students, we will going to keep this group of a cell, this group of a cell or a callus into one medium. We will provide the sufficient amount of a nutrient to it, we will provide the sufficient amount of a hormones to it, where the proper growth and the differentiation take place. When I said growth, it means the cell division process take place, but when I said the cell differentiation, that will help in dividing the specialized kind of cells or the tissues. So at first we will keep this callus into that medium, into the where we will going to provide the hormone which help in the cell division, and then we will keep it in that medium where the cell differentiation can take place easily. Now, due to which there is a formation of a plantlet, and these plantlet is then we will take it out from the medium and we will place it in the soil. There this again starts growing and it becomes a mature plant. So we can say that the plantlet which is placed in the soil, they will help in the growth of the mature plant. Okay, so this is how we can use with the help of the tissue. See here, this is also a kind of a asexual reproduction. How we have taken a tissue, we have taken a specialized cell, group of cells, and then we had provided a medium to it from outside. Normally what happened, we know that the fusion take place and it is present in the ovary itself, and there the cell division occur after the formation of the zygote. Then it will get converted into embryo. Here we are separating the cell and the tissue, we are providing the medium to it, and due to which the growth happens, the division occur. Now when the division occurs students, then it will continue to grow into an embryo, formation of an embryo which we called as a callus, and then we will going to provide the hormones to it. So again we are artificially we are giving environment to it, right? So when we will give the environment to it by providing the hormones to it, then it will grow into a small plantlet. Now this plantlet we will going to soil, we will going to plant it in the soil, and they will grow into a mature plant. Now the question arises, why we are doing so? Why? What is the need of a tissue culture? Okay, there are different plants, they used to grow from the different parts of the plant, that is one thing. But why we are artificially doing this, separating the tissues and growing the plants? See, the good thing about the tissue culture, that it is basically help in making a plant disease free. The tissues which is taken outside, it will first of all it will be it undergoes a kind of changes in which we will make it as a disease free condition. Even we can say that we can provide it the sufficient nutrient that will make it a disease free plant, that we will going to provide the resistance to that of the plant. And this technique, it is also is important for the, or we can say that this technique is important since it help in providing resistance to the plant regarding the diseases. And this is basically used for the ornamental plant. Okay, now see here, the next one is the spore formation. So one of the other plants which used to show this asexual reproduction is the spore formation. See here, in a many simple multicellular organism, specific reproductive part can be identified, and the thread like a structure that develop on the bread above are the hyphae of the bread mold, and they are not the reproductive part. On the other hand, the teeny blob on the stick structure are involved in reproduction, and the blob are sporangia, which contain cell or spores that eventually develop into new rhizopus in individual. Basically, see students, in a simple multicellular organism, there are specific reproductive part which can be identified, like in case of a fungi. What happened in the fungi? Fungi are of a different type, students. One of the fungi is there in which you will find that there is a thread like a structure is there that basically develop on the bread. You have heard about bread mold. What happened when you kept bread for a longer period of a time? So due to the presence of the moisture, it will be attacked by fungus, right? So we can say here that attacked by the fungus. So what kind of a fungus it will be? So it is the mold. Okay, that fungus is mold, that's why it is called as a bread mold. So what happened there, you will see that when you take that bread mold and keep it under the microscope, you will find that mold is what it is, the long thread like a structure, the and this which we also called as hyphae, and this hyphae of the bread mold, it consisting of a bulb. As you can see here in this structure, this is the spore formation, and when you look at the structure, students, you will find that it consisting of a long thread like a structure, and blobs are there on the stick of the structure, and that is involved, that help in the process of the reproduction. Actually this blob is called as a spore. These blobs are the sporangia, and these sporangia consisting of the spores in it. Okay, so sporangia, it is present in the fungus, in the mold, and which is just above to that of the or we can say the hyphae, and which is just above to that of the hyphae, or which is just above to the stick structure, which is involved in the reproduction. What is the role of the spores? You can see here, student, this is what, this is the sporangia, and this is sporangia, it bursts, and due to which small spores are coming out of it, right? So these spores are, what is the role of these spores? Actually these spores, it eventually developed into a new rhizopus individual, and the spores, they are covered by a thick wall that protect them until they come into a contact with another moist surface and can begin to grow. Basically, you know that they need to reproduce. So what happens, suppose you had kept the bread in a dry condition, so that will not going to affect it until and unless you will provide a moist surface to it. So when a moist surface is there, then the fungus will attack. It means the fungus grows. So then the development of the fungus, the reproduction of the fungus take place when the condition is what, it is moist for them. What happened when the condition is moist? At that time, the spores which is present in that of the on the hyphae of the rhizopus, that will grow, and that will develop this rhizopus, which consisting of a blob like a structure on it, that is called as a sporangia, and this sporangia consisting of a spore. So when the condition is good, this sporangia, it bursts, it releases the spore from it, and the spore will again get developed into the rhizopus. So this is how without showing the sexual reproduction, the asexual reproduction can be seen. Okay. Now let us see here, students, if we talk about the asexual reproduction, we are knowing that this mode of reproduction is what, it is shown by the single organism. It means it involves a single organism where one of the parent will going to perform the process of a reproduction to give birth to their to give birth of their own kind or identical to them, because they are what, they are genetically identical, that we have already studied about it. Now see here, there are a lot of organisms which used to show such kind of a reproduction, that is asexual reproduction. I already had explained to you about the asexual. A means no, and sexual means the sexes which is involved in it. So since no sexes are involved in it, that's why it is a asexual reproduction. Now see here, there are some of the single cell organism are there, especially unicellular organism, which used to show such kind of a reproduction. Now let us see here a unicellular organism which shows the kind of a reproduction which is called as a fission. Why the term is fission? So in class 8 we have already we are very well known about the binary fission, students, but here it is not written as a binary fission, because the fission can be binary fission or fission can be a multiple fission. So For unicellular organisms, cell division or the fission leads to the creation of a new individual. Many different patterns of the fission have been observed. Many bacteria and protozoa simply split into two equal halves during the cell division, and in organisms such as amoeba, the splitting of the two cells during cell division can take place in any plane. See here, when I said about the fission, what is a fission? Let us try to understand. Fission means what? Breakdown. It means we can say that a unicellular organism, when it splits or when it divide to form two new organism or more than a new new organism, they are called as the fission. That is the process is called as the fission. Fission in Hindi, this is the breakdown only. Okay, so splitting of the body into two or more, that is called as the fission. And fission is of two type. Binary fission is there and multiple fission is there, students. Different kind of organism, they used to show the different unicellular organism, like amoeba is there, paramecium is there, lesbania bacteria is there. These all are what they used to reproduce by the binary fission. But some of them, like that of the plasmodium, used to show the multiple fission. Now let us start with the amoeba. Here it is written that in organism such as amoeba is there, the splitting of the two cells during division can take place at any plane. At any plane, what does that mean here? That this is what? This is that of the fission that is occurring into that of the amoeba. So here you will find that ultimately the same amoeba get split into two. So what happened? How amoeba shows the process of a binary fission, students? Basically, the amoeba has what? When it reaches to the maximum size of the growth, then first of all the nucleus, as you can see here, the nucleus elongates. Okay. So the elongation of the nucleus occur, as you can see here, students. Yes, along with it the size is increasing, but the first one you can see the nucleus is elongating. So when the nucleus elongate, and finally afterward what happened? This nucleus gets split into two. During the elongation or during the depression which is formed in that of the nucleus, the cytoplasm also elongates, the body also elongate, and finally when the nucleus, first of all you can see the nucleus get elongate, it get divided into two, and then there is a formation of a depression occur between that of the cytoplasm, and due to which it will get split into two amoeba. Okay, two identical daughter cell. So if we talk about the amoeba divide into two part. One part, each of the part is what? It is surrounded by that of the nucleus. Now here one more thing that I want to explain, students. This is the first one. This is what? This is the first one. It means this is the parent amoeba. This amoeba, it grows. First, what happened here? As you can see, yes, as you can see here in this picture, the first one, what? The amoeba is there, and this amoeba, the nucleus which was present in it, it starts elongating. Right, and due to this elongation, students, there is a depression formed between the nucleus, and due to which the nucleus divided. Now the last one, this two which is formed by this one, so this is what? It is a parent cell, and this is what? This is the daughter cell. This is the parent cell and this is the daughter cells. So from one parent cell, how many daughter cells are produced? That is 2, and it is because of that this fission is called as the binary fission. Since one parent is dividing into 2, that's why it is called as the binary fission. Now see here, we can say that the daughter cell are what? They are not similar, but they are identical to their parents. Why is it so? As you can see that these two daughter cells are formed by this parent cell, it means the same nucleus is divided into these two daughter cells. So the genetic material, the genetic information that is present in this nucleus, it will be given to these daughter cells, and this will be what? It is exactly the same what a parent have, and that's why the daughter cell which is formed here in this the daughter cell which is formed here in these two is what? They are having exactly the same same characteristic, or they are genetically identical to their parents, and or we will not going to say similar, but they are genetically identical to their parents, and that's why we can say that they are identical to their parents. Okay, they are now similar, there is no such variation you will find, they are exactly the same copy, exact copy of their parent. Okay, so the one parent cell divided into two daughter cell, and such kind of a fission is called as the binary fission, students. Now let us move on and see the other thing. However, there are some of the organisms are there, they are somewhat more organization of their bodies, such as seen in Lesmania, which caused the Kala Azar, and which have a whip-like a structure at one end of the cell, and in such organism binary fission occur in definite orientation in relation to these structure. Other single organisms such as the malarial parasite Plasmodium. Yeast on the other hand can put out small bud that separate and grow further. So yeast is there which used to produce the small bud that gets separate and it grow further. Now let us see the Lesmania, students, because it is said that in case of a binary fission in amoeba. So what is there in the amoeba? Amoeba shape is what? The fission that take place, the binary fission, it take place in amoeba also. The binary fission take place in the Lesmania also. So in that case, students, you will say that in case of in the amoeba, since the shape is what? That is not regular, and that's why the production that happen here, the formation of the nucleus occur, it's irregular only. But in case of a Lesmania, which is what? Which is a protozoa, students, it forms a disease that is called as Kala Azar, and this will get divided in, first of all, you can see in this picture itself, it consisting of what? Is consisting of a nucleus. Then the C, if you look at the second, in the B structure, you will find that the cell is what? It is little bit elongating. And in the C structure, in the C portion, you will find that it get divided, it get divided into two nucleus. In D, they get separated from each other. So the formation of the in the first structure, that is the normal Lesmania, and the second one you will find that there is little bit elongation you will find in the nucleus. In the third one, they get divided, there is a depression is formed between that of the two nucleus. In the fourth one, you will find that both of the nucleus, they get separated from each other. It means the proper formation of the nucleus has already been taken place. And in the E one, you will find that now the body starts dividing, and this time the body always divide vertically, students, okay, up from downward to upward, but yes, vertically. So the division happening here, and that's not irregular but in a proper manner, where the body is dividing vertically into two. Here at the F1, you will find that the body get properly divided, and each of the part consisting of the nucleus in it. Finally, both of them will forms into or develop or get separated to each other to form the two daughter cell. Okay, so in case of a Lesmania, the binary fission take place, but that is little bit different from the binary fission that take place in the amoeba. So yes, students, in both of the cases, the one thing common here, that a one parent give rise to two daughter cells. Another factor is there, that in case of a binary fission, like we have seen in case of a amoeba, what is the difference that you will find in case of a parent cell and that of the daughter cell? So the size Yes, the size of the daughter cell is what is smaller than that of the parent cell. In case of a lesbian, it means this daughter cell, it will further grow and develop, and when it becomes fully mature, when its size becomes it expanded, only then it will start showing the binary fission, students. Although this process does not take very much time, but yes, the binary fission, the daughter cell produced here, it will first of all, it will grow and mature or developed into a fully matured amoeba, and then it will show the process of a binary fission. Similarly, in case of a Leishmania also, here also the division occur. So first of all, the nucleus divided and the cytoplasm divided, and then it will get divided into two, and here again the two daughter cell which is formed by the parent cell, it will grow, it will develop when it become fully mature, and then only it will become fully mature, and then it will again show the process of the binary fission. So this is what, student, binary fission.